What's up, guys? Today we're going to go over model organism studies by me, myself, and I, Gavin Yarbrough. A model organism is a non-human species that is widely studied in scientific research because it is very easy to manipulate and study in the lab. It has a relatively short life cycle and shares important characteristics with other organisms. Model organisms are used to study biological processes, test hypotheses, and develop new treatments for diseases. Examples of model organisms include the fruit fly, the roundworm, the zebrafish, and in our example today, mice. Model organisms are nothing new. The use of model organisms in scientific research has a long and rich history. In the late 19th and early 20th centuries, scientists began to use a variety of organisms, including fruit flies, mice, and bacteria, to study genetics and other biological processes. These organisms were chosen because they were easy to grow, had relatively short life cycles, and produced large numbers of offspring, making them ideal for genetic studies. One of the first model organisms used in genetics research was the fruit fly, Drosophila melanogaster, which was first used by the geneticist Thomas Hunt Morgan in the early 1900s. Morgan and his colleagues were able to use fruit flies to study the inheritance of traits, and their work helped to establish the field of genetics. In the mid-20th century, the mouse became another popular model organism, particularly for studies of mammalian genetics and development. We will talk about mice more later on. As the 20th century progressed, more species were introduced as model organisms to be used for research, bringing us to today, where a wide variety of model organisms are used in research, including bacteria, yeast, nematodes, zebrafish, and many others. These organisms continue to provide valuable insight into the fundamental processes that govern life, and they are instrumental in advancing our understanding of human biology and disease. But that's enough talk about genetics. Let's look into how model organisms have high flexibility in terms of their usage. Due to model organisms having a small size, short generation time, high reproductive rate, and well-characterized genome, and similarities to humans, they can be useful in a variety of different research. My favorite example is the roundworm, which was used to study aging and neurodegenerative diseases by identifying mutations that affect these processes and using the worm to screen potential drugs for treatment. Other examples of modern organisms being used for very specific research that are not on the slides include the fruit fly being used in evolutionary biology research to study speciation, and mice being used to study addiction. In the now, let's observe how researchers from UNC's Lab of Cell Bio and Physiology use model organisms. Mice mayhem! UNC researchers elected mice as their chosen model organism to study the effects of e-cigarette exposure on the fertility, pregnancy, and offspring of female mice. Specifically, the study was designed to determine whether e-cigarettes, which also contain nicotine, are any safer than traditional cigarettes for pregnant women. The researchers conducted a series of tests to study the effects of e-cigarette vapor on the fertility, implantation, and growth of mice. The researchers split the female mice into two groups, a control group where the mice were exposed to air, and the experimental group consisted of mice who were exposed to e-cigarette aerosol consisting of nicotine, propylene glycol, and vegetable glycerin for 3 hours a day with two 2-second puffs per minute. The study found that exposure to e-cigarette vapor delayed first-time pregnancy, affected fetal survival, caused smaller litter sizes, and a delay in embryo attachment. Female offspring exposed to e-cigarettes in utero also failed to grow to a normal size. Model organisms are what made this experiment possible. Although I mentioned many different reasons towards the beginning as to why model organisms are advantageous in the lab, ethics become another concern. An experiment like this could never, ever be done on humans, as it would be wrong and very harmful. By using model organisms, we can further our research at a faster rate by not only being able to manipulate them so easily, but rely on their fast reproduction rate in the case of fatalities. Not to mention, over 95% of the mice genome is similar to that of humans, making them an ideal model organism for studying human conditions and genetics. I hope you are just as excited as I am to see where the model organism research goes over time. The possibilities are endless,